Sure. Sure.
Okay, so today you're done with the first drawing, right? And then after this one, we discuss, and then we're going to look at our tissues. We're going to finish cytology and histology, and then we're going to go to the next. She said we can go there and look at our slides. You're going to start looking at your slides, start doing your histology drawings. Or I can, maybe after the break, I can move the microscopes here so that I thought she doesn't have class there. So we finished looking at the cell, we finished looking at all the organelles. We need to find out so again, again, you say to yourself, hi, my name is Robert. I am a human being, right? Living organism made up of cells. And all these cells that I have, they're all busy. All my cells are alive, and they're all busy maintaining what? I wanted you to remember that word, homeostasis. Remember that all our cells, individual cells, 75 to 100 trillion cells, are always being exposed, no matter what you do, right? You hear the alarm clock, you upset the homeostasis already. The glucose level in the blood, when you wake up, it's down, so you get hungry. You see, it's always exposed to ever-changing environment, so it's busy. And we look at the different organelles that make up this cell. You see all this, the pretty cell, all of this, the parts of the cell, they all have a job, right? And you notice, or I hope what you realize is all these different parts of the cell, they're there, they have a job, and their main goal is to maintain homeostasis. Now when you look at any cell, you know that you think of it in terms of what is the composition of that. I know it has salt, it has oxygen, carbon, all of those ingredients for life. You miss one, it's impossible to maintain life, okay? So now that we formed the cell, there it is. What is the fate of the cell? What happens to a cell once it is formed? Did we discuss this?
Yeah, it either dies.
This is where we ended, right? There are three things that can happen to the cell, right? It will either die,
Stay
and you
in
know the
interphase.
definition of apoptosis, necrosis, right?
Yep.
It can stay alive, or it will divide. If it stays alive, so this is the entire life cycle of the cell. So once it's formed, right here, it can just stay in this phase, or it can just immediately die. Remember the difference between apoptosis and necrosis. It can just stay right here, and the cells that stay in this phase, they're like they don't grow up, like guys, right? They never grow up. They're like children forever. So you have the red blood cells, the nerve cells, and your muscle cells. So once they're formed, they just remain in this phase called interphase. or they will divide. So any cell that is not dividing, it is still alive, it is metabolically active. It's still doing the day-to-day activities, maintaining homeostasis. What events do you think is going on in interphase? Muscle, it must be making muscle proteins, right? So it can contract. Red blood cells, it must be making hemoglobin so that I'm absorbing hemoglobin, releasing oxygen. You know, it's still busy. How about your neurons? It must be making neurotransmitters, right? Or receiving information. So you see, it's busy. Even skin cells. Any cell that is not dividing, it stays here. It's not dead, like your bone. Your bone is not dead, but it's here in this phase if it's not dividing. Like your skin cell right here, right? When it goes up, it's dead. But the skin cell that is not dividing, is it not doing anything? No, it is busy making proteins. Okay, so that's why we have to discuss protein synthesis. In your book, there is a nice page in your book that I like it. It step by step discusses protein synthesis right here. You see the focus. First, there's a nice figure 3.34. There's a nice scheme of the process of protein synthesis right here. And then if you go look at the next figure 3.35, it's showing you how the protein is made. And finally, the focus figure 3.37, the whole process of transcription and translation. So I want you to look at that one, okay, when you have the chance. But here is how is protein synthesis, what is that event, okay? So this is, another term for protein synthesis is called genetic translation, right? Genetic translation. Every protein in the body, let's say hormones. So you just ate. After any meal, we know that the glucose level in the blood is high. Do you follow so far? Right? I had my breakfast. I know after I digest, I absorb it. Glucose level in the blood is high. So, do you know of insulin? Right? We're all familiar with insulin. That's our hormone that controls the sugar in the blood, right? If we have problems with insulin, we become diabetics. Insulin is not just magically present in the body. You make it in response to the glucose in the blood. Okay? It's not anything in the body is not just there. You have to make it. So the pancreas will have to make that insulin. And how do they make that? Protein synthesis. Just like your hair or your skin contains keratin. Does it make sense? Keratin is protein again. Do you think your skin cells already have that? No, it has to make it. Or, hello there, like your egg cells, if you did in vitro fertilization, right? So there's that cell. You introduce the sperm over there. So there's a nick over there. How are you going to fix that? You need to repair that, right? You need to, when there's a leak in the roof, you have to replace that. You need to repair that. How will the egg cell repair that? Protein synthesis again, okay? So that's why we have to discuss the process of protein synthesis, which is very easy. It's really not that hard. Where are we going to start? You need to know these two steps. The two steps in protein synthesis are transcription and translation. So this is the picture in your book, that focus translation, figure 3.37. Is it 3.37? Okay, so look at this one. Let's just, for example, we need to make insulin because I just ate. Okay? So this is my pancreas. The islets of my pancreas have to make insulin. Okay, it's not present. It's not, you just make it on demand. Everything in your body you have to make on demand. So here, Where is the code? It's in the nucleus. Anything you need, the nucleus, the genetic code, you remember your DNA, the DNA codes for everything that you need. So let's say this is the part of the DNA that codes for that one. So you see the nucleotides A, T, C, G. So let's say it's over here. That's the code for insulin. You're going to make a copy of that code. That's the first step. The first step in making any protein or protein synthesis is transcription. What happens in transcription? You're making a copy of the DNA code. Again, why is that important? Because DNA does not leave my nucleus. It's very precious, right?
Right.
Can't go in and out, because you damage your DNA. And so that copy is called mRNA. So what happens during transcription? Transcription produces an mRNA, which is the copy of the DNA. Okay? Where is this event happening? Inside the nucleus. Okay? Just like your digestive enzymes, to digest anything, you have to make the enzymes. These are proteins. This is how they make it all the time. So look at how they base pair. The DNA nucleotides, in the past, I have had students make a copy, make their own mRNA, the code, and sometimes I do it, sometimes I don't. So just be prepared. So the DNA nucleotides are A, T, C, G, right? A pairs with T, T, C, TA, CGGC, right? You know this, right? Because you took Bio 175. But then the RNA has no T, right? It has uracil. So that's the only one that's missing with RNA, right? So look at how the RNA is formed. A, you have U. You see that? How it base pairs. And so this RNA strand is an exact copy of the code for that insulin hormone, okay? And this is the one that will leave the nucleus. If you look at this slide and if you look at your book, you just read it, that figure, that page, or step by step it tells you. So once you form the mRNA, there's an enzyme that makes this. It's called RNA polymerase. You don't need to know that, but look, the mRNA, which is the copy, is the one that's able to leave the nucleus. Where is it going to go? You guessed it? Yes, very good, the ribosome. It sits on the ribosome. Look, there's the mRNA with the nucleotide strands. So the DNA carries the code, the mRNA carries the codon. You see the word codon? Okay? So, and every triplet codes for an amino acid. So every 3 will code for an amino acid. Are you following so far? So it sits on the ribosome and it just waits. In the meantime, in the cytoplasm of every cell, you have the amino acids there. All the amino acids, the 20 amino acids, are present there. They are bound to another type of RNA, this one. It's called tRNA. Okay? So this tRNA, again, the tRNA bounds every amino acid that we have in the cell. It's just in the cytoplasm. And so the tRNA, look at this tRNA over here. It has 2 ends. One end binds the amino acid, and the other end has the triplet. It's called anticodon. It's just the matching triplet that base pairs with the codon of the mRNA. Are you following? This is just a review of Bio 175. You got that? So look at whichever tRNA is able to base pair with that mRNA. That's the one that will bind or base pair with the triplet on the mRNA. You see that? And then on the other end, at the other end of the tRNA is the amino acid. So as it base pairs, you see, it just starts forming your polypeptide. So here in the cytoplasm, you translate the code to an actual protein. Do you think you got this? What do you think? One more time. Whatever protein I need, right? Enzymes, keratin, collagen, whatever it is, you have to make it. It's just not all present in your body. Your cells have to make it on demand. Okay? And so they need the recipe. And the recipe is where inside the nucleus. The DNA carries that recipe called the DNA code. And so there will be the first step, there will be a copy made of that code because you only have one code. You can't just keep bringing it in and out. You just take a picture like we all do with our smartphones. Take a picture. A lot of people are so spoiled with that one, right? You just take a picture and then you go out here and that picture is called the mRNA. It's an exact copy of that one, base pair, okay? So you should be comfortable with pairing. In the past, in the test, my past test, I will write, given this specific code, DNA code, I'll just use different base pairs for the nucleotides, so let's say A, T, whatever. And then I'm going to write, please give me the mRNA, the resulting mRNA. So you just base pair, right? And then give me the tRNA. You need to find out where the first site of translation happens. It doesn't just translate anywhere. There is what you call the start codon and the stop codon. Again, once I have base, this one, no problem, right? So let's say this strand, this is what I'm going to give you, the DNA. You're just going to start making a copy. You just copy, base pair, and you form the mRNA, if I ask you that one. For the tRNA, you need to find out where you're supposed to start. There is always a start codon. The start codon is AUG. This is the start codon. How do I remember? I remember that classes in this country start in September. So I just write August. I don't know. So that is your start codon, August. So, and so, no problem. DNA, I give you the DNA, you write the mRNA. Okay? And then my next question will be, write the complementary tRNA, taking note of where you're supposed to start. So here, you're going to look for where AUG is, and that's where you start translating. You're translating now to the actual protein. So this, you see this one, methionine? AUG codes for methionine. So that the protein strand always begins with methionine. Okay? So just look for that. And then look at this one. It will just keep on coding, encoding, encoding until you get to a stop codon. So once the codon here, the mRNA strand, once the codon consists of UAA or UAG or UGA, it stops. It will no longer code for that. Because why? These three codons do not code for anything. So it ends there. So when it's done, this dissociates, and you have now the protein floating in your cytoplasm. You have translated your protein. You follow? One more time. There it is. I need to make an enzyme, let's say pepsin, because I just ate steak. So the first step, transcription in the nucleus. The copy is called mRNA. It base pairs with the DNA, that segment of the DNA. And this mRNA leaves, goes to the ribosome. The ribosome is another type of RNA. It's called rRNA. But anyway, it sits there and it just waits for the complementary base pairing from now tRNA. And tRNA, on the other end of the tRNA, is the actual amino acid, and that's what you want. So it will start base pairing with the mRNA codon. Where will it start? It looks for AUG. That's where it will start. As the part of the mRNA that it will start base pairing. And it's methionine. And it will keep on until it hits the part of the mRNA that gets to the ribosome, until that part is either UAG, UAA, or UGA. When it gets here, it's done. It's finished. And so the ribosome releases the mRNA strand, and this dissociates. You now have the protein that you need. This was in the ribosome. Next step, this goes to the, what does your note say? Rough endoplasmic reticulum. That's when you modify the protein, remember? And then from the rough endoplasmic reticulum, Golgi body to be shipped out or used inside the cell. Okay? So you see the difference between transcription, translation. Where is the location of transcription? Nucleus, very good. Where is the location of translation? Cytoplasm. Are both events intracellular?
Yes.
Yes, very good. Just want to know if you know your terms. Intracellular, extracellular, right? Okay? So what phase in the life cycle of the cell is this going on? Interphase. So is interphase a dead phase, a stationary phase where there's nothing going on? No. This is what we call metabolism. They're always busy. Okay? They're making proteins. And that's the bulk of the life of any cell, protein synthesis. And that's why in some schools, they take forever. This RNA, by the way, the one that is formed is called a pre-mRNA, because some of that one, there's the nerves. It's not the actual strand that is going to be translated. So you have to, before it even leaves, some of that mRNA that's formed, you have to put the introns and the exons. But I feel that sometimes it's too much for our brain cells. It's just the basic of protein synthesis. To me, that's good enough. As long as you understand. You need to know which one happens in the nucleus. So now you see translation. Now you know what translation means. This is also referred to as genetic expression, right? You're expressing your genetic code. Protein synthesis, genetic expression, that's all the same. And this is what we take advantage of when we, pharmacologies or pharmaceutical companies, this is what they do. They take advantage of the bacteria, they harvest. You know how they formed the penicillin? It's a product of bacteria, right? And so that's how they do it. They do, it's called recombination. So they tweak the DNA code, they express, and they form all the different kinds of medications, or they form different kinds of bacteria that can fight the pathological bacteria. So you can use this slide or this slide. This first is the transcription. So these first two slides before the final slide, I just did this, or I incorporated this because it's more detailed in case some of you like detail. So you see first in the nucleus, you make a copy, and then it leaves the nucleus. This is the nuclear pore over here. You see that there's the nuclear pore, so this mRNA will go to the cytoplasm, and in the cytoplasm, there, sits in the ribosome right here. Look at the start codon is AUG. So it will just look for that one, and this is your tRNA, and that is methionine. So that's always the first one that base pairs, and it just keeps base pairing and base pairing. So the two amino acids will be joined by peptide bonds until, look, it just becomes larger, until it gets to what? The stop codon, which is UAA, UAG, or UGA. So if you don't remember the stop codon, if you remember the start, look at UAG. It's just, you jumble the words, right? UGA. And then UAA. Okay? So like I said, in the past, I had students do that in their test. Sometimes I don't, sometimes I do. So just, oh, I forgot. Okay, so if they're not busy, if they are not in interface right here, they must be dividing. So right here. And as far as cell division is concerned, there are two general types of cells. The sex cells and the somatic cells. The sex cells divide by meiosis, and the somatic cells divide by mitosis. There are only two types of sex cells. Your what? Eggs and sperm. Egg cells or oocyte, and your sperm cells. So it's not the vagina, right? It's not the cells of the penis. No. It's just the sperm cell and the egg cell that divides by meiosis. Again, in the past, I had like a matching type. Mitosis, meiosis. See both, D none. So I just randomly pick a few cells. You tell me if it divides by mitosis or meiosis. Again, if you don't see the word this one or that one, it must divide by mitosis. Okay? But whether it's mitosis or meiosis, it consists of these phases: prophase, metaphase, anaphase, telophase. The test, especially in your lab exam, you should be able to identify a cell that is in interphase, prophase, metaphase, or anaphase. So what is the goal of cell division? The goal of cell division is to produce two or more cells, right? If it's meiosis, the product of meiosis is 4 cells. If it's mitosis, you produce 2 cells. So you're always doubling up, right? And this is how we keep our skin. We always shed our skin. If there was no cell division going on, then we'll lose our skin, right? So, how do you produce 2 cells exactly alike? It must be that before this mitosis even proceeds, what do you think? I'm thinking I must double up on everything. What do you think? You must double up on everything so that the 2 cells that's produced, they look exactly alike, right? Okay. So right here, look at in interphase immediately. if you're going to if you're going to divide, you have to make a copy of everything. This is where the centrioles is very important, right? Cells with no centrioles will not divide. So I know that I'm going to double up on all my organelles. I want to look at my chromosomes. You want to preserve the human count, which is, how many chromosomes do you have?
23.
Huh?
23.
23 pairs, right? So, so each of our cells have 23 pairs. Are you familiar with the word diploid? Diploid is any cell that has two sets of chromosomes. We have two sets, one from my mom and one from my dad. So my cells are diploid. Or you can also write it this way, 2N, right? Okay, I have mommy genes and daddy genes. So that from my mom, I have 23. From my dad, I have 23. Okay? And so I said, or we discussed, that the first step, before cell division can even proceed, you must double up on everything. So the first step in any cell division is doubling up. As far as DNA is concerned, you call it DNA replication. So that you will end up with, how many is this one? 92, right? So I have 46 times 2, I will have 92. You following so far? So that when I divide, I'm going to have two cells looking exactly alike, two diploid cells, same chromosome count, 46, that's human cell. Okay? So look at this process of DNA replication. How are they able to make an exact copy? The first step is you separate the two strands. There's an enzyme called helicase that separates that. You don't need to know that. But that's how you look at what happens. You separate the two strands and you just base pair another DNA strand. So what is formed when you form this one, when you form the copy, this is the copy. They make one copy and another copy, where each one will have an original strand plus a new one, old and new. So that's why the replication is referred to as a semi-conservative replication. You don't just keep all this DNA and make a new one over here. You separate them, and that's how you base pair. Okay? Semi-conservative replication. I want you to know the difference between chromatin and chromatid. Chromatid and chromatid. D for division. So, if this was a cell that is not dividing, just in interphase, the chromosomes are referred to as chromatin. So each chromosome, right, is made up of two strands, complementary strands. You call each strand chromatin. But once it's going to divide, you refer to the strands chromatid. I don't know why they're very specific. To me, they're all the same. But they're very specific with terms. So just in case they ask you that or it comes out in your board exam, now you know the difference between the two. When you see the word chromatid, you know that this cell is dividing. When you see the word chromatin, you know that this cell is just in interphase. Okay? So first step, I double up on everything, right? Now I'm ready to divide. So what are the steps in, let's assume that this is mitosis. What are the steps? Prophase, metaphase, anaphase, telophase. Look at this one. Look at the difference between the cell in interphase and a cell in prophase. Look at the chromosomes here. It's lighter in color, right? This one has only 46. But when it doubles up, it becomes 92. You can see it's darker. So under the microscope, I will have a slide on mitosis. You scan around, look around, and look for a cell in interphase. When the nucleus becomes so dark and prominent, you know that the cell is in prophase. One more difference, this cell in prophase eventually, look at it. Here in interphase, the nuclear pore is very intact. But in prophase, in addition to the chromosomes doubling up, so being prominent, you see that the nuclear pore starts disintegrating because they have to separate. So that's one more clue if you want to know if the cell is in prophase or interphase. This nuclear pore, this blue one right here, it starts disappearing because you have to separate the chromosomes. So here, what do you call the chromosomes? Chromatid. Here, chromatid, dividing. D for dividing. Okay? Okay? So, what are the different phases? You don't need to tell me what are the events. Look at this one. So there's your prophase. See, this one is being separated. There's a cell in prophase. And what's the next one? Once you make a copy, they meet in the center. This one set and this one set, they meet in the center. It's almost like they discuss, oh, this is what you're supposed to do when you go there, right? So they discuss and then they meet together in the center, they discuss, and then they start being pulled to each end. This is your centriole. The centriole forms the microtubules, and that's the end of the microtubule attaches to the kinetochore of your chromosome. You don't need to know those terms, but then they start being pulled to each end. So here, the two copies, they meet in the center and they start being pulled to each end. So when they meet in the center, that is metaphase. I know you know that. Okay? And when they start getting pulled to each end, anaphase. What's here? This is the 23 from daddy, 23 from mom. 23 from dad, 23 from mom. Until you get to the end, telophase, where very distinct you will see the cleavage plate or cleavage furrow. So when you see that, that's telophase already. And cell division does not end until the last step, which is called cytokinesis. Cytokinesis is the separation of the two cells. Cytokinesis. Cyto for cell. So look at this one. This is how you should be able to label a cell in prophase, metaphase, anaphase, telophase. For the lab exam, I will have a few microscopes. Of course, most of them will be histology, but there'll be one or two that will be on mitosis. So I'll say identify. So you're going to write a cell in prophase. So maybe I'll say assume that this is a somatic cell. At this point, there are how many chromosomes? So how many chromosomes at this point?
Still 96.
96? 92?
92. I can't add.
92, right? And then maybe I'll ask, what is the ploidy of this ploidy? Diploid or haploid? Diploid, right? Look at mitosis. So when they divide that way, right? And at the end of mitosis, you're producing two cells with you reform the individual 46 chromosomes. So with mitosis, are you following? You start with diploid, you end with diploid. All the way it's diploid. Whether it's prophase, metaphase, anaphase, all of these cells, if it's a somatic cell, these are all diploid cells. They all have mommy and daddy genes, all of them. Okay, except that some of them will have 92, some of them will go back to 46 interphase. Look at this. I know this is interphase, very light in color. I know this is now prophase. Look, it doubled up. This is 46 chromosomes, this is now 92 chromosomes, right? But this is still diploid. Do you know the difference now between diploid and haploid? We didn't get to haploid yet, but diploid is if you contain both mommy and daddy. Any cell that contains both mommy and daddy is called diploid. So even if it's 92 over here, I don't care about the count. I will call it diploid because it contains both mommy and daddy genes. Okay? Okay? So let's say another question. So identify, you're going to write telophase. Okay? So maybe I'll ask, some cells will never, ever have this stage in their life cycle. Name two. You're going to answer red blood cells, your muscle cells, and your nerve cells. You will never see a red blood cell or a muscle cell or a nerve cell in telophase, in prophase, in metaphase, anaphase, right? Because why? They don't have centrioles. So they will never ever have this phase. They will only be permanently in interphase. Okay? Maybe I'll ask here, identify, you'll say telophase. At this point, there are how many cells? It's still one. Right? It's still one. It didn't completely separate yet. So it's still one. At this point, what is the chromosome count? It is still 92. It didn't completely separate yet. Maybe another question will be, this process is an entirely intranuclear event. What do you think? Is that true or false?
False.
Yes, that's right. It's false, right? Because
Repeat the question?
entirely nuclear. Is it entirely nuclear? Must not be entirely nuclear. Very good. Why? Because when it separates, you're going to form two cells. Okay? So, but is it entirely intracellular? Yes. All of these events inside the cell. Okay? Let's talk very briefly about meiosis. You're going to visit meiosis again when you take up the reproductive system. But with meiosis, are you good now with somatic cell division, mitosis? Okay? Meiosis is the same. You see the pictures? The same. It looks, a cell in prophase looks like this. Metaphase looks like that. There's no difference under the microscope. The only difference with meiosis, so you have your egg cell, right? Your oocyte or your sperm cell. These are the only two cells that divide by meiosis. So you start with diploid also. This is the parent egg cell and the parent sperm cell. With meiosis, the way the cells divide, the egg and the sperm, instead of dividing that way, they divide this way. You see? So there's, again, what are the phases? Prophase, metaphase, anaphase, it looks like this still. Except that the cells that's produced, look. So there's the difference. You see? you're producing 2 cells. After meiosis 1, I'm producing 2 cells. Look, what is the chromosome count? The answer is 46. Except, look at this one. It contains half. Maybe this is the daddy genes plus the copy. And then the mommy went on the other side plus the copy. So, chromosome count, yes, 46. But, haploid. That's what you mean by haploid. Haploid is any cell that contains half the set of chromosomes. And this is just half, right? This is just from my mom. This is just from my dad. Plus its copy. Okay? And meiosis is a 2-step cell division. Immediately after this one, it divides again. Another set of, looks like this again. Prophase, metaphase, anaphase, telophase. Looks like that. And so then, finally, you produce 4 cells with 23 chromosomes each. Okay? So you see the difference in chromosome count after mitosis compared to after meiosis. You see that? So how many cells are produced by mitosis? 2. How about meiosis? 4. What's the chromosome count after mitosis? 46. Diploid. What is the chromosome count after meiosis? 23. Haploid. Is this human? No. This is not human, right? When do you reform the human organism? During fertilization. So when let's say there's an egg, this is an egg, and when the sperm comes and dumps its 23 chromosomes there, you reform the human. That's the only time we call it human. So that's why if there's no fertilization, don't we get rid of it in our menstruation? So you get rid of it. It's not human. Haploid. Haploid. Okay? So you only use the word haploid in meiosis. But how does it look like? It looks like this. So again, in the lab exam, I'll just say, identify. You'll say, cell in anaphase. And then maybe I'll ask, assume this is a sex cell. Or maybe I'll say, assume this is a sperm cell. How many cells will be produced at the end of this process? 4. Right, 4. Very good. Then maybe I'll ask, the cells produced by sex cell division will have how many chromosomes in each? 23. Okay? Okay? And you will understand the process in more detail when we study the reproductive system. But for now, that's good enough for you. You should be able, again, I'm not going to ask you to give me the events, what's happening here or here, but I want you to at least be able to tell me and train your eyes when you look at it under the microscope. This is cell division. This is prophase. This is metaphase, anaphase, telophase. I always have practice exams too. I notice that students, when I do practice, I use practice histology exam on, I just flash pictures and you answer. They do better when it comes time for the actual histology lab exam. Okay? So do you have any questions? That is the life of the cell. That is what happens to every cell. It's either once you form it, now you know. You've been introduced to every cell in your body. You know now the composition of every cell in your body. You know now what that cell is doing. It's either in interphase or it's in cell division, mitosis or meiosis, or it's on its way out, necrosis, apoptosis. Now we move on to histology. So two or more cells make up a tissue. Okay? So break for 5 minutes. Then we start with histology.